I think we are good to go if everybody's happy. So we're going to go through, we'll spend about 3 minutes for each of the talks. It's quite quick and we'll save the questions for the end. And Anna Strampelli is going to be my co-moderator and take some of the questions and help direct them back to the speakers at the end. And then we can open it up for a general discussion on anything that people would like to discuss related to the main session and anything that we've seen today. So I would like to introduce our first speaker, if I can work out how to use my slides. There we go. Okay, so our first speaker is Awosolu Oluwaseun, and he's going to be speaking to us about trends in falciparum malaria prevalence, parasite density, and associated risk factors in Akure, Southwestern Nigeria, and implication for malaria elimination. Take it away.
Thank you very much, Andrew. I'll be speaking on trends in falciparum malaria prevalence, parasite density, and risk factors in Akure South local government area of Ondo State area. Actually, malaria is a disease of major public health problem, right? Particularly in Africa. It's caused by parasite of the genus Plasmodium. Malaria is transmitted by Plasmodium infected Anopheles mosquito during blood meal. So the major of this study is to determine the prevalence of falciparum malaria with respect to demographic characteristics, for example, age and gender, and also socioeconomic characteristics such as occupation. The methodology involves the collection of 2 to 3 ml of venous blood from patients and aliquoted on Workman filter paper and also put in EDTA for both microscopy and also PCR analysis. Thick and thin smear were actually prepared from blood in EDTA for microscopy analysis and then stained GIMSA, and the thin film was fixed and viewed under light microscope in order to determine the use of Plasmodium, and it was eventually counted with the help of the thick smear. Also for the PCR analysis, filter paper blood spot was cut into 1.5 ml tube using a puncher, and the genomic DNA was actually extracted using Cayenne blood and tissue kit following the manufacturer instruction. The eluted DNA was kept in -20 degrees until the PCR analysis, and Plasmodium was identified with the head of nested PCR using genus-specific and species-specific primer according to the method of Snolo Assay 2002. And table 1 actually the result of malaria prevalence with regards to gender, age, and occupation. With regards to gender, male actually had the highest prevalence of 69.5%, which is higher than female. And also with regards to age group, less than or equals to 12 years of age, which are children, actually had the highest malaria prevalence of 77.0% compared to other age groups. And with regards to occupation, farming had the highest malaria prevalence of 86%. So all these are actually in line with many publications that have been published previously. So with this research, it is obvious and we can conclude that malaria remains a major public health problem in Akure South local government area of Ondo State. Thank you very much.
Drew, you're on mute, I think. We can't hear you, or at least I can't.
That was my fault, yeah. So that was excellent. Thank you very much. You get the time, and we'll move on to the next slide. So we have Dr. William Garrood, who's at Imperial College London, and he's going to speak to us on assessing off-target effects of CRISPR-Cas9 based gene drives in Anopheles gambiae. Should I take it away, William?
Thank you, Drew. So for CRISPR-Cas9 gene drives, it is important to understand how specific the cleavage of CRISPR-Cas9 is and whether there can be cutting at genomic sites other than just at the intended on-target site. So to assess whether such off-target editing was able to occur within a mosquito gene drive, we utilize the following pipeline. So firstly, there is an in vitro assay to predict where these off-target sites could be. This combines a large quantity of mosquito genomic DNA and is incubated with Cas9 and the guide RNA of interest. The analysis is then conducted to identify where the Cas9 has cut, and this is done by the presence of the DNA double-strand break, and then this produces a set of genomic location that can be cleaved in vitro. So the second step is then to use this map to then perform deep sequencing of these predicted off-target sites and assess whether we can detect evidence of CRISPR-induced mutation. So the top right here shows the output from this off-target detection pipeline. The sequence is on the left of the output from the in vitro assay, and it gives a list of sites and then shows the top sites that could be cleaved in vitro were closely related to the guide RNA of interest. So this means that they had 3 or fewer mismatches compared to the on-target sequence. So by then sequencing all of these genomic sites shown here across various different gene drive strains, all 3 of which utilize the exact same guide RNA, we were able to detect evidence of CRISPR-induced mutation at 5 of these genomic sites. This is indicated by the red boxes, and this was in comparison to the wild type strain. So the detected editing frequency was always very low. We never saw greater than 1.5% of the reads from the amplicon sequencing that had mutations at the corresponding target site sequence. Furthermore, the editing could only be detected in a gene drive strain where Cas9 expression was regulated by the VASA promoter, which is not tightly restricted to germline expression. When using a different promoter for regulation of Cas9 expression, ZPEG, which was germline restricted, we no longer saw evidence of CRISPR-induced mutation at these predicted off-target sites. So in summary, CRISPR-Cas9 can cleave at genomic site other than its intended target site in the mosquito. However, by sensible design of the guide RNA to limit closely related site and tight temporal restriction of Cas9 expression to the germline, can reduce off-targets to minimal levels within the mosquitoes.
Excellent timing and very good talk. Thank you very much, William. So for our third talk, we have Jacques Nambani from the IRSS in Burkina Faso. He actually works alongside Etienne Bilgo and Abdoulaye Diabate. And his talk is going to be heritable cultivable bacteria associated to wild Anopheles gambiae sensu-lato in Burkina Faso for paratransgenic control and perspectives for malaria. Is Jacques in our audience? It's a shame I don't see him. Okay, I'm going to move on and perhaps we can go back to this later. So the fourth speaker is Rose Joseph, and I can see you there. Hello, Rose. And Rose is from the University of Namibia, and she's going to be talking to us about the evaluation of neonicotinoid insecticides against pyrethroid resistance Anopheles gambiae sensu-lato in the Zambezi region of Namibia.
Thank you, Andrew. Well, pyrethroid resistance was confirmed in 2020 in all malaria endemic regions of the country. And in order for our vector-borne control program to establish an effective resistant management plan, it is imperative for the program to establish the susceptibility of local malaria vectors to novel insecticides, which are said to have a different mode of action towards pyrethroids. Well, this study aimed to investigate the susceptibility of Anopheles gambiae sensolato in Zambezi region to both Fludora fusion as well as Sumishal insecticide. The study was then conducted in Sibinda village, which is northeast of the country. This was done by collecting larvae in their natural habitats and rearing them at the insectary to the F1 generation. And then 3 to 5 day old female mosquitoes that were only fed on 10% sugar solution were then used for their assays. Mosquitoes were exposed to Fludora fusion, which was coated in 250 ml bottles and also exposed to WHO tubes, which was lined with Sumishield impregnated papers. As per the findings of the study, it showed that Fludora fusion induced 100% mortality of Anopheles gambiae mosquitoes, which was 24 hours post-exposure, and Sumishield only induced a 98.75% mortality, which was evaluated 7 days later. In conclusion, Anopheles gambiae mosquitoes in Zambezi region are susceptible to both Fludora fusion and Sumishield, and this could possibly be because of the unique mode of action and the absence of cross-resistance to other classes such as pyrethroids, and this could possibly be included in the indoor residual spraying campaigns to control Anopheles gambiae sensolato in Zambezi region. And since the study was preliminary, it is recommended that it will be evaluated across all malaria endemic regions of Namibia, and further molecular analysis will also be done of the species. The study was done in partnership with the University of Namibia, the Ministry of Health and Social Services, as well as Clinton Health Access Initiative and Bayes. Thank you.
Thank you very much, Rose. Excellent. I can see that Jacques has joined us, so perhaps if I can go back one slide. Jacques, are you able to present your slide now? Perhaps. Okay, well, maybe I'll give you a second to see if you can get your mic and camera sorted out. And when you're ready, maybe let Anna know with the message, and we can go back. So for this next talk, we have another Jacques Delon, who is from the Fondation Congolaise pour la Recherche Médicale. Forgive my pronunciation. And he's going to discuss the prevalence of non-falciparum malaria in individuals living in south of Brazzaville and its surroundings in Republic of Congo. Jacques, are you able to present? We can't hear you yet. You might need to unmute yourself. Well, I can't see Jacques here either.
I thought I had at the beginning of the breakout session, but
yeah, so do I. I saw him right at the beginning. Maybe he lost connection. Perhaps. Okay, well, I'll move on and we can go back. So for the final talk of this session, but hopefully not if we can go back to our other lightning talk speakers, we have Rebecca Rosenberg, who joins me here at Johns Hopkins University actually, and she's going to be presenting an exciting new technology called VectorCam, which is a low-cost handheld tool for automated rapid morphological identification of mosquito species. After you, Rebecca.
Thank you. Yes, my name is Rebecca Rosenberg, and I am the project lead for VectorCam at the Johns Hopkins Center for Bioengineering, Innovation, and Design. Vector surveillance is the process of analyzing the population characteristics of mosquitoes in an area in order to make informed decisions toward elimination of vector-borne diseases such as malaria. To begin the process, mosquito specimens are collected at rural sentinel sites. They're collected at rural sentinel sites across the country using techniques such as CDC light traps, human landing catches, and spray catches. These sites are often sparsely distributed across a target geography, but due to resource constraints are treated as representative of much larger areas. The collected specimens are then transported to a central laboratory by vector control officers and analyzed for characteristics such as species, sex, abdominal status, and sporozoite presence. It's crucial to understand these characteristics because they provide insight into the behavior of vectors in those areas. For example, while two different species of Anopheles mosquitoes might be able to transmit malaria, mitigation efforts will be different for species known to bite indoors versus outdoors. This information is then used to inform intervention decisions, such as distributing bed nets or indoor residual spraying at the highest level. Our group is working to automate specimen processing based on the model used in Uganda. Our recently published algorithm is capable of identifying up to 39 species of mosquitoes with high-resolution imaging equipment and has been trained on a first-of-its-kind database of over 100,000 images. Currently, our team is working to deploy an updated, lighter version of the algorithm on locally available Android smartphones and using cost-effective imaging software. The system, which is a low-cost, high-throughput entomologist in your pocket, will allow a minimally trained worker to take a single image of a mosquito specimen, which will then be processed through our several algorithms to make determinations of species, sex, and abdominal status. Pursuant to this goal, our group designed several prototypes and recently took them to Uganda and Ghana for preliminary feedback from partners at the Ministry of Health, President's Malaria Initiative, National Malaria Control Division, and Makerere University. We received overwhelmingly positive feedback as to the potential of the technology for automating and task shifting vector surveillance. Additionally, through co-creation with vector control officers, village health teams, and technicians, we determined that future priorities should include higher throughput, working space within the field of view, and operation without internet connectivity. With this feedback in mind, we've developed several new prototypes, which will be the subjects of a formative usability study planned to take place in Uganda and Zambia in January 2022. By automating the process of mosquito identification, we allow for lesser trained individuals to participate in vector surveillance, thus enabling timely and cost-effective expansion of programs in partner countries. Thank you very much.
Thank you. That was excellent. So I'm just wondering, do we have any of our other speakers here? Maybe one of the Jacques. If you're, I can see you in the list of participants, but maybe you're having trouble. Are you able to speak up? Okay, that's a little bit of a shame, but I'll move on. So before we get to the discussion, I'd just like to highlight a couple of things. So one, which I've been asked to highlight, is that there is the Keystone Symposia coming up on April 10th to the 13th in 2022. And this is going to be at the Beaver Run Resort in Breckenridge, Colorado. And the symposia is Malaria, Confronting Challenges from Drug Discovery to Treatment. And we would encourage people to sign up and consider joining this symposium. And the second thing to consider is World Malaria Day 2022, which is on April 25th. And again, there are going to be many activities run by Johns Hopkins University. We would encourage you to keep up to date with everything that's going on. And Johns Hopkins always runs their own one-day event, which is always fantastic. They have world leaders giving a number of fantastic talks. So with that, I think I'll open it up to some discussion, and perhaps Anna, maybe you had something that you'd like to get us started with.
Yeah, I did have some questions actually for the speaker. Should we go in order of presentation? So maybe I might start with Awosolu, unless anyone else has a question they'd like to ask first.
I would say I would encourage people to speak up, and you might be able to use the raise your hand feature or any of these things. So this is open mic night now, a little bit different from.
But an informal session, I think. So do speak up. But I'll start with the first question. So Awosolu, thank you very much for your talk. It was really interesting. And I was wondering if you could tell us a bit more about what the current control measures in the area that you conducted your study are and how they relate to the results that you find. And also if you had any recommendations that you would make based on your results on how better to control malaria in this area.
Yeah, thank you very much for that beautiful question. That's nice. Actually, they provided mosquito bed nets together with some collaborating companies. We have malaria control program using mosquito nets. However, there is need for education for people to be able to use this appropriately, because it is not being properly used in those areas. So if assuming it is properly used, I think this will help. And aside from that, mosquito net does not cover all the time. There are other times when you cannot use mosquito net. For example, you can use it during the day when you are not actually sleeping, when people have gone to their various places of work. So that could also cause some certain problem, or we have to use other means of control measure. Maybe we have to use kind of repellents on, or something we can actually wear.
Right.
I think that day-biting mosquitoes constitute a problem, since you can see that with the farming occupation, of people who spend time outside, the prevalence is a lot higher than, for example, for civil servants who presumably spend more time inside. So definitely, I think that.
Yeah, exactly. So if we need something to protect, if you're on the farm, things like that. Yeah. Perhaps people who stay late during the night doing their business.
So despite all my years working in bear research, I still find it very shocking to see something like 77% prevalence among children under the age of 12. So how many of these infections are symptomatic?
I didn't check for asymptomatic.
But perhaps you have an idea, I think.
Because well, I previously worked on that in another area, another hospital. But for asymptomatic, I think is more. Yeah, because virtually everyone is exposed to mosquito bites. Yeah. So if you are bitten maybe twice or thrice, then you are likely going to have parasite that may likely survive eventually, and then you may have the same. Many people are actually, over time, you might have immunity, which may not necessarily make the infection become heavy, such that you want to, it's kind of eating. So, but if you test the individual, such person might be positive for malaria, but not really feeling the effects all the time. Yeah, except maybe when you are stressed up, you are weak, and then the infection comes up.
Okay, excellent. We had one hand raised in the audience. Would you like to pose the question yourself, Adebanjo?
Thank you, Andrew. Yes, one of my questions has been raised, just to know whether the sampling, where the sampling was taken, is it hospital based or is among the general population? But I think you will move in that direction to ask whether we have asymptomatic or symptomatic malaria. Yeah. The only question that I would just add to this is maybe we just need to know which among the people tested positive for malaria, which of them tested positive based on microscopy, and which of them tested positive based on PCR test? What's the prevalence for PCR test? Then what's the prevalence for microscopy?
Yeah, thank you for that beautiful response. Actually, this analysis I put here on the table is for microscopy, because I was able to do the parastemia calculation, the parasite density calculation through the microscopy. So I decided to use that here, and the prevalence for that is 64.89%. But for PCR, I have more than that, which is I have about how many percent? That is, I have 405 individuals that were positive for malaria instead of 390 for microscopy, then 405 individuals for PCR analysis. And that is about 67%. Yeah.
Yeah, it's always difficult trying to work out the best technology, whether you have a high technology or low technology that's more applicable in the field. So I think in the interest of time, we have lots of questions coming your way, but perhaps people can ask Oluwaseun some questions in the chat, and perhaps there are some for our second lightning talk speaker.
Actually, I think that Jacques is ready to speak. It's in the chat. Yeah.
Okay. Which Jacques? Jacques with Etienne. So this one, this heritable cultivatable bacteria.
Hi, Andrew.
Hi there.
Thank you for all. And I'm going to talk about heritable cultivable bacteria associated to wild Anopheles gambiae SL in Burkina Faso for paratransgenic control perspective of malaria. Mosquito and bacteria association is well established. And this point, how much we know about them in mosquito swarms. To answer this question for new tool development for malaria control, we investigated the role of microbiota. We investigated the role of microbiota on mosquito swarm. And this we collected couples and single mosquito. In the lab, we dissected the reproductive organ and crushed them in the tube and plated this crush on 2 medium at 37 degrees. 1 to 2 days after, the microbiota colonized was morphologically identified, and then the genus or species of bacteria was screening using Biotech2 compact system. According to our data on microbiota abundance number per mosquito reproductive organ from swarm, we reported that with the couple male test, there was more microbiota than with a single male one. And we reported also that there are anti-malaria paratransgenic bacteria and biopesticidal microbiota. And this biopesticidal microbiota was higher in single male mosquito test than coupling male mosquito. Thank you.
Thank you. That's really interesting. Thank you, Jacques. So I think we'll carry on with some of the questions that came up earlier. And so Anna, was there something else for one of the other lightning talk speakers that already come up? You're muted.
Sorry. I have some questions. I hadn't seen any on the chat, so maybe I can ask a question to Will. Will, I was wondering if you could just briefly expand on what the CRISPR-Cas9 gene drives that have been developed do and why it's important to avoid these off-target effects.
So yes, the gene drives have been developed to generally target female fertility genes, then we reduce the to essentially lead to a population crash. So because each generation, the way the gene drive construct is homed is through the cleavage of CRISPR-Cas9 at your desired on-target site. You don't want to have any off-target you want to have very minimal off-target cleavage, because then otherwise every generation, if it could cut elsewhere, it has that potential to introduce new mutations, which you ideally don't want. So it was important to show, firstly, that we could actually detect these mutations if they did happen, and then secondly, show that actually by optimizing the gene drive strategy, you could reduce these mutations to a level below the minimum detection threshold, and so then you'd be less worried that there was, this potential for mutation to occur while the gene drive was spreading.
So William, just an extra point on that. If you did have off-target spreading alongside a gene drive, do you see that as a big issue that should prevent gene drives from being used?
No, because it's, even if you get off-target editing, it doesn't mean that this is directly a problem, because most edits will occur intergenically, so they won't have a direct cause. They'll just be lost from the population through natural selection. So it's not really a case that if you see off-target edits, then it's a massive problem. It's just you would prefer not to see them. But yeah, you wouldn't expect them to do something drastically bad.
So would there be a sort of strategy that would first look at the potential impact of these off targets before something goes to the field?
So you could possibly, if you had certain genes, for instance, which you would not want to see off targets in whatsoever, say for instance, genes involved in insecticide resistance or vector competent, then you could possibly run an assay to look just at those and see whether there were any off targets that would occur there. I mean, if you've got off target there, then it would possibly be a problem, but you would have looked at this probably much earlier on in development. So you would have found it quite early on.
Okay, excellent. Unless anybody has any other questions for William right now, I might be tempted to throw one to one of the other speakers. Any questions? Okay, so I actually was very intrigued by the vector cam, and what I'm imagining in my head is like these new apps that you have where you can point them at plants and it tells you that it's the wild East Asian oak tree from somewhere or another. And I think that these things are absolutely fantastic. And I was wondering, you said that right now it's based on internet use, and is this so far been a problem in field sites? And is this something where you can take the photos in a field site and then later, when you have a better internet connection, analyze those photos once you're at a site with a stable internet connection? Is this really a big issue?
Yes. So the part of this that kind of requires the cloud connectivity is if you want to run sort of a really large algorithm, it's very computationally intensive. And so originally our 39 species algorithm is very large and would need to be run on the cloud. And so images taken locally would need to be uploaded, run on the cloud, and then the information transmitted back to the user. What we're trying to do now is create a lighter version. So maybe with only a few of the most important species that will be able to actually run locally on these available Android smartphones, so that there's no internet connection that's necessary. Because especially at some of these very rural sites where they're doing vector surveillance, having internet is just not possible.
Okay.
And sorry, go ahead.
I was just going to ask, like, it seems that the accuracy is actually very high, but I was wondering, would there be an option to say take several photos of the same insect specimen and have a higher accuracy?
Yes. So our algorithm is actually trained we've taken about 3 to 5 images of each specimen, and that's the way that it's trained. However, that's supposed to help it. It just adds more to the data set so that it has a better chance of doing well on one image of a single mosquito. That is possible. Technically, that could make the accuracy better, but it would slow down the users pretty significantly, which is a cost-benefit analysis.
Okay. Anna, did you have something to ask?
Yeah, I did have a question. I was wondering whether VectorCam is the use is just for identifying the different mosquito species, or if it's kind of seen as kind of an information database that grows with each photo that is taken, and whether this kind of map that you would create is kind of accessible to the users independently for the photos they take, or whether kind of all the photos will feed into this one big map of surveillance, if that is even is one of the uses of VectorCam.
Yes, so more than the images themselves, what is useful is obviously the species information, as well as some of the other things we're working toward. So the sex of the mosquito and the abdomen status. So the goal is that that information would be made available on, like, just like you're saying, like a large public map, so that the individuals who are making the strategic intervention decisions have as much information as possible to make those decisions more specifically for each area.
Okay, great, thanks. It's really so, but for now, it's the app is based on identification of what it now does is identifying the mosquito species. It doesn't yet have this database application. Okay, cool. Does anyone else have questions for Rebecca, or should I ask a question? I have a question for Rosalia, but I'm wondering whether anyone else has a question for Rebecca. Awosolu, I think you have your hand up.
Yes, actually, I have a small question. Thank you for the wonderful. Actually, I just want to know, how long is it going to take to identify a species of mosquito by this particular app? And what is the basis? I mean, what is it based on? The identification, is it based on information you already put in the system, or actually?
So if I understand your question, you're asking about how our system identifies a mosquito?
Exactly. How long?
Yes. So we use a convolutional neural network, which is just a type of machine learning model that is able to, so we've trained it on a very, very large number of images, so in the hundreds of thousands. And we actually are not quite sure at this point exactly what it's looking at on the mosquito, but it's sort of meant to be a black box. And so there's more analysis that can be done to try to learn what the computer is actually using to make those determinations, but generally that is not known.
Okay, thank you very much. So how long does it take to identify a single species of mosquito?
Just seconds.
Oh.
You just snap the picture and it processes in, yes, seconds.
Okay, so I just tried to download the app now. I downloaded Vector app. Is that the same app?
No, it's our app is not publicly available right now.
So okay, great. So let us know when it is available so we can use to identify our species.
Absolutely.
We also have a question in the chat for Rebecca,
Sure.
which is, what's the accuracy from Zeph Nelson? What's the accuracy percentage? Can the technology identify sibling species, especially in Africa where many such mosquitoes exist?
Yes, so we don't have that information at the moment, but that is something we're very actively working towards, especially for the gambiae sibling species, where we have some very preliminary proof of concepts that suggests that it could be possible to use this same technology to identify within the sibling species, but we don't have accuracy information on that quite yet.
Thank you. And I think also Adebanjo, you have your hand up.
Yes, my question is similar to the one in the chat, that has it been tested for intervention? Has it been tested in any environment for malaria intervention or to eliminate malaria mosquito, to get malaria species in, maybe with the aim of eliminating malaria mosquito in a particular environment or particular region? Has it been tested?
So we did take prototypes to Uganda and Ghana in August of this year. So we've done some initial field testing, and some of that was just meant to capture images that we can use to continue to make it better. But yes, we're looking most closely for in Uganda, for example, at Anopheles gambiae, Anopheles fenestis, the main vectors of malaria in that area.
Thanks.
I think, Rebecca, you're getting lots of questions. Another one in the chat from Stephen Panosian. So one, does the algorithm also take location into account? And two, are you considering expanding into other genera?
So to question one, so the algorithm itself does not consider location. It just looks at the image itself. However, we do plan to collect alongside species data, location data as well, so that can be kind of all placed on a map appropriately. Question number two, I'm not quite sure I understand.
I think so you have, because you're, I guess that you're only looking at Anopheles mosquitoes, or are you also looking at other genera?
Oh, no. We have our original 39 species algorithm includes Anopheles mosquitoes, Culex mosquitoes, Aedes mosquitoes. Yes.
Great, thanks. And there was a question whether the mosquito can be damaged, but I guess that
yes,
it won't be released again. So
yes, if the mosquito itself, our original algorithm was actually trained almost exclusively on images of mosquitoes that were wild caught, desiccated, and some missing appendages like wings or legs. So yes, that is something that it does still work.
Sorry, I'm just reading a question in French and trying to translate it. Give me 1 second.
That's okay, thank you.
I'm sure we have some bilingual people who can easily translate this in our audience.
It's just a long one, that's all.
Okay, perhaps Etienne or Jacques.
Yes, I think it's just asking a question that was already asked before, whether whether your algorithm differentiates between highly related species of mosquitoes, because they can be morphologically very similar, but I think you said it does.
The sibling species, so we're working toward that. At this point, it does not. Yes.
Oh, okay, cool. Thank you.
I'm just reading from the chat now. The capture technique, we've used all different types. CDC light traps, we've used spray catches, we've used human landing catches, all of them. Show key characteristics. No, that it uses.
Yeah, this is one of those black box situations, right? Where with
Yes.
machine learning, it's very difficult to know exactly what is being picked up on.
Yes, exactly.
Right. Still, it's very cool. It'd be good to get inside the brain of that black box.
Yes, and that is an analysis that we are also working toward. It's quite difficult, actually.
Yeah. Okay. Well, I was supposed to close this session at around past, and we've gone 50 minutes over, but it has been very, very exciting. I think everyone's been very motivated, and we've had a very good day. I am very impressed by all of the talks today, all of the talks in all the main sessions and the lightning sessions. And I can't thank you all enough for joining and making this a very wonderful experience. And it is very exciting to be around the future of malaria control. So many younger scientists who are going to be pushing us forwards. So I think it has come time to close the session. And I will bring up again just these 2 points, that there is the Keystone Symposia, which is happening at Breckenridge, which is malaria confronting challenges from drug discovery to treatment. And this is happening in April. And shortly after that symposia is World Malaria Day 2022, and Johns Hopkins will have a day-long event for that. And I encourage you all to sign up and to follow the Johns Hopkins Malaria Research Institute on Twitter. They often announce lots of these different events. So thank you to everybody, and I wish you all the best for wherever you are, morning, afternoon, or evening.